All right, so today we will look into the role of cells in our organisms and what is the smallest unit of living cell. So, a cell is known as a building blocks of an organism and is also known as a basic unit of life. But why is it called so? See, a cell, when they come together, they make tissue. Tissue is actually made up of cells which perform a specific function or a particular function. Now, this tissue, which is going to perform a particular function, forms a organ. Now, organ is incorporated into a system which is known as a organ system. Over here, I have given an example of digestive system. There are different kind of systems, which is one of them is respiratory system as well. So here the digestive system consists of a mouth, a esophagus, you have small intestine, you have large intestine, and together they make up a whole system. So this is known as an organ system. Now, when different kind of organ system comes together, they form an entire organism. Now, this entire organism wouldn't be possible if there was no cell in the first place. So hence, if no cell, no particular tissue, no organ, there will be no system and there will be no organism. So hence, these cells are known as the basic unit of life. Now, moving on, there are two types of cell. Mainly, there is prokaryotic and then there is eukaryotic cells. Prokaryotic cells can have an example of bacteria, and eukaryotic cells have examples of animal, plant, fungi, algae. Now, why they are called so? Pro means old and karyon means nucleus. So these type of cells are having old type of nucleus. You can guess what it means by eukaryotic cell. Eu means new or true, and karyon again means karyotic. So these prokaryotic cells are having old type of nucleus while the eukaryotic cells are having new type of nucleus. Now, when you look at the genetic material of a prokaryotic cell or the bacteria cell, you will see it's just lying inside of a cytoplasm. There is nothing surrounding it. But when you look at the eukaryotic cell, it's having a nucleus which is actually surrounded by a membrane. This right here is the DNA, that is the genetic material, and it's going to be surrounded by a membrane. Here, the nucleus is surrounded by a membrane, so we will call it a nuclear membrane. So, right over here, this is the membrane which is surrounding the DNA, and we will call it the nuclear membrane. Membrane is nothing but an envelope. Think of it like the genetic material is caged inside of something. But the prokaryotic cell lacks the cage. It means that it lacks the membrane. So it's free. And this over here, the DNA is going to be known as nucleoid. So that's what they are called in prokaryotic, but in eukaryotic, you're going to call it a nucleus. All right, now let's move on to a bacteria cell. We'll go from the outer surface to the inner surface. Now, the first thing you see is a flagella, which is like a hair-like projection coming out of the body of the bacteria, which is meant for locomotion or just movement. Most of the bacteria can have one flagella, or they can even have multiple of them projecting out of the body. So let me just draw that structure for you. Here it is, like they just have one flagella, just like this, or they can have multiple one. Now, the second thing we're going to see over here is actually the pilus. Pilus is in some of the textbook. It is also given as fimbriae. They are in the AP biology textbook. It is said that both are the same thing, but they are slightly differing in function. So pilus is a bit longer than the fimbriae. They both are hair-like projection coming out of the body. Pilus is only meant it's only going to work when they have to exchange a genetic material during conjugation, that is the reproduction of the bacteria. So here I'm telling you how it is much more larger than the fimbriae. Fimbriae is much more smaller than the pilus. So it's just used for genetic exchange during conjugation or reproduction, while fimbriae can only be used to attach itself to the host cell. That's it. That are the two jobs of pilus and fimbriae. Now, moving on to the next thing, we are going to see that, let me write it down for you, there's attachment of host cell. Now, moving on, we are going to see the next thing, that is a capsule. Now, the capsule over here, it is only a slime-like thing that is around the bacteria, which is going to help it prevention of dehydration. So I'm gonna write it down for you. It's having a slime-like thing around its body, and it's going to prevent dehydration. All right, so next thing that we see is here is the cell membrane, which is also known as a plasma membrane. Membrane helps to separate the cell from the other environment, the outer environment, and is going to also help in exchange of substances in and out of the cell. The next thing over here that we see is a ribosome, which is embedded in a cytoplasm. Now, cytoplasm is just like a jelly-like fluid in which all of the organelles are present. For example, in bacteria, there is genetic material, cellular DNA, and chromosomal DNA, ribosome embedded in the cytoplasm. Moving on with the cellular DNA, here it is actually having no nuclear membrane. At the down, you can see I've put a picture of a eukaryotic nucleus, which is having a nuclear membrane. But here in the bacteria, it is lacking the membrane. Hence, it is known as a nucleoid region because it's not having any kind of membrane. In eukaryotic, you would call it a nucleus, but in prokaryotic, you're going to call it a nucleide. Now, there's also a cellular DNA, which is known as plasmid, present in the bacteria, which is not shown over here, but we'll discuss about it later. It is actually very useful for us. Now, the next thing is the cell wall. Cell wall in bacteria is made up of peptidoglycan. Peptido means there is like a chain of amino acid that is present, which is known as peptide. Now, over here, there will be a chain of amino acid or proteins, and glycan is just sugar or carbohydrate. So, the cell wall will be made up of structure of sugar and protein in a chain structure, that is peptidoglycan. So, cell wall also helps in maintaining the shape of the cell and it's meant for protection as well. Now, moving on to plasmid. Plasmid is actually a cellular DNA present in most of the bacteria. Not all of them are going to have it, only some of them are going to have it. Now, what is the basic concept behind it? When there is a antibiotic that is given to a bacteria, certain antibiotic is given to a bacteria, these bacteria who are having a plasmid, they contain genes which are resistant towards the antibiotic. That means they are not going to die, they are going to sustain themselves, they're going to live. Hence, this plasmid is meant for survival purposes, and this is also used in genetic engineering when we think about useful stuff that we can do in the medical field. So the basic concept is plasmid is used for survival, which is actually passed down to the other bacteria during conjugation process in reproduction. And that's it for cell structure and prokaryotic cell.